Hey guys, today me and my friend we are going to talk about another section of genetics, rather a very interesting one, which is karyotyping and karyogram. I'm sure you guys must be familiar with these terms, but let's get deeper into it. To start with, let's talk about karyotyping. So basically, karyotype is the number and type of chromosomes present in the nucleus. Basically, it is a complete set of chromosomes of a species. And it is mainly used to determine the gender of an unborn baby. Cells are then harvested from the fetus and then they are photographed to be visualized. So basically, the photograph that you get as a result is the whole process of karyotyping. Next, we will be looking into the methods of karyotyping, basically how it's done. So there are two main methods that are used, and the first one is the chorionic villi sampling. And as mentioned in the name, it involves removing a sample of the chorionic villus, which is basically the placental tissue, and it is done by inserting a tube via your cervix, as seen in the first picture above. Even though it can be done at almost around 11 weeks of pregnancy, but there is a slight risk of miscarriage, approximately 1%, which is considered to be the only drawback of this method. The next one is the amniocentesis. So, amniocentesis involves the extraction of a small amount of amniotic fluid, which contains your fetal cells, and it is usually done with a needle. And this method can be conducted at around almost 16 weeks of pregnancy. And again, it has a risk of miscarriage, but a very slight risk of miscarriage, around a 0.5%.
So now we move on to karyogram. Karyogram is a diagram of the chromosome of the species, as we can see here on the right. And the chromosomes are stained, so that's why they're visible. And they are arranged into their homologous pairs, organized by their sizes, from the longest one to the shortest one. And here in this karyogram, we have the human karyotype, which consists of 22 pairs of autosomes and one pair of heterosomes. And therefore, we could say that a karyogram shows the karyotype of a specific species. So with a karyogram, we can identify the gender of the unborn child by looking at the heterosome we have seen earlier. An X and a Y combination will have a baby boy. An X and an X combination will have a baby girl. Simple as that. Moreover, it can test for chromosomal abnormalities. One of them is aneuploidy, which is the presence of abnormal number of chromosomes. Usually in humans, we have 46 chromosomes, while in the case of aneuploidy, we can have 47 chromosomes, 45 chromosomes, and so on. Here's an example. And as we can see, there's two X chromosomes which coded for a girl, but however, there is a non-disjunction occurs in the 18 chromosomes. And non-disjunctions happens when a pair of homologous chromosomes or the cystochromatid were unable to separate properly during anaphase 1 and anaphase 2, which result in cells with unusual number of chromosomes. Some are missing and some are having additional one. These non-disjunction can potentially lead to many genetic syndromes, and the one we're seeing here is the Edwards syndrome, aka trisomy 18. Tri means 3, 18 just means the 18 chromosomes. These syndromes affect the growth and development of the child, and normally baby will die shortly after being born. A karyogram can also test for chromosomal translocation and deletions. Here in this example, we have a deletion in the short arm on the 5th chromosome. This picture shows that a part of the chromosome, which contains genetic information, has gone missing. And this situation is often known as Cri-du-Chat syndrome, means cat's cry. The patient who diagnosed with this syndrome have a high-pitched crying, sounds like a cat, so that's why it was named like that. The patient with Cri-du-Chat will have developmental delays, language and physical difficulties as the result of the missing part of the chromosome. Just to clarify that non-disjunction can occur in any chromosome, not just the 22 pair of autosome, but also in the pair of heterosome, which is the sex chromosomes. And this could lead to, for example, trisomy X, which is 3X, or even tetrasomy X, 4X, and even pentatromy X, which is the 5X here in this picture shown. And this is a summary of the vocabularies in our presentations. You can take a screenshot of it or write down some notes. It will be very helpful, and thank you for listening. Thank you.